Take the introduction part and the last part will taken by our partner Rathea from Cambodia and Bonnie from Philippines. So now our topic is in different canopy types induced soil microfauna variation in bloom nature research. Okay, before that model conceptual, we know that abundance, the diversity of the soil fauna was estimated to 23% of the total diversity of living organisms, and the huge numbers illustrate that the importance of the soil microfauna as the engineer of the environment, and is affecting the below and above ground environment. As we know, in the different succession stage, there has different plant community composition, and in basically in the same climate condition, the factors determine the soil microfauna distribution and the abundance. More depends on the soil, the physical and chemical properties of soils, such as the soil water, soil pH, and the organic matter, and so on. And it also depends on the litter quality and quantity, such as the litter quantities, carbon, and the nitrogen rate. And previous, the information on the soil microfauna distribution and abundance most collected from the agro environment and ecosystem. So where the management practice alter the soil properties and the plant compositions. So there is no much more attention to the different canopy forests. So our aim is to focus on that three questions. The first one is, how does variation in soil microfauna occur in the different canopy plots? And the second one is, how does species abundance varies in different layers of soil? And the third one is there a relationship between termites and earthworm distribution? So the method will
We conduct our study in Mekong, the library. We selected 9 plots in the Bloom nature research here. And the red dot here stand for the closed canopy, the green one stand for the open canopy, and the yellow stand for the open land. In each plot, we already do the random sampling to select 3 out of 9 subplot, and we do the sampling size 2 sampling in each subplot. And then we do the soil macrofauna. We sort our soil macrofauna according to the layer that we designed, like the first layer from 0 to 10 centimeters, second from 10 to 20, and the third from 20 to 30 centimeters. During the soil sampling, we also collected data related to the environmental factor, like infiltration rates, soil moisture contents, and basal area. For the data analysis, we use GLM to see the significance of the soil macrofauna to the factor affect them, like different type of canopy and soil layer. And we also use the CCA to see the significance of the environmental factor on the species abundance. Yeah. And the next slide, our group mate Bonnie will continue.
Okay, So after sorting the soils looking for macrofauna the whole day, so we counted 1,921 individual soil macrophonas, which belongs to 22 orders in 10 arthropods classes. And among these individuals, termites comprise more than 50% of the whole populations of the soil macrofauna, followed by the ants, centipedes, beetles, and earthworm, which only 18%. The rest for the other 17 orders. And then for our statistical analysis and other data manipulation, data analysis, we focus on termites, ants, centipede, beetles, and earthworm as our response. And then this figure shows that based from the stock bar, there are different the distribution and based on abundance distribution of each of the 5 most abundant species in each habitat: open land, open canopy, and closed canopy. And it also shows how species are distributed in different layers. It also shows that in open land, the species are distributed in each layer. There's a minimal distribution, and then in open canopy, there's unequal. And here we can see in the closed canopy, first layer has differences, high difference in second and third layer. And then based from our ANOVA analysis, we computed there is high significance in this for these orders in habitat and layer, and we combine the interaction between habitat and layer. So there's a very high significance. And then this table shows termites does not influence the distribution of earthworm, because we computed no significant results in whether the termite affects earthworm distribution. And then in this study, we also want to know what are the factors that affect why there are variation in this soil macrofauna abundance. And then we also try to see the 3 factors: soil moisture, infiltration rate, and basal area. They are also significantly different in different canopy types. And then we try to interpret these factors which affecting the orders. So based from this CCA plot, so the 3 factors: water content, basal area, and infiltration rate, doesn't affect the distribution of taxons. The red ones are those taxons that we identified. And then discussion: in open canopy land has higher macrofauna abundance than other canopy types. This is contrary to what we predict, because mostly, not all, because we use the canopy as factor. We predicted before that the closed canopy forest should have a higher species abundance, but our results shows that open canopy has a higher abundance. Maybe the reason is behind the numbers within you see the termites. Termites has very high numbers compared to the other taxon, and then we found them in open canopy mostly. And then the abundance of the soil macrofauna favors topmost layer because of ability of food for them. So yes, based from other studies, the soil macrofauna favors the upper layers because the abundance of nutrients and other food available for them. And then the distribution of termites might not affect earthworms, yes, but we're also thinking of, depending on the degree of termite abundance, there are areas where we find a large number of termites, and we don't see earthworms. And when this number of termites is only small, we found earthworm. So we need to, and we also need to see our sampling areas is very limited to see the factors that affect termites and earthworm abundance. And then, as conclusion, soil macrofauna abundance varies in different canopy plots, with open canopy having the most abundant among plots, and also the same first layers being the most abundant, and the frequency of termite does not influence earthworm distribution. Although we also therefore conclude that although there is a significance between the environmental factors to different canopy types and taxons to different canopy types, but the relationship between the environmental factors and taxons doesn't have a good relationship. And then we would also like to take this opportunity to thank all the sponsors this course for giving us knowledge and opportunities. And special thanks to those persons who helped us in the field data gathering and data analysis. So, thank you.
Thank you. Questions?
Can we go back to your results slide? First, so, there's a lot of information on here, which is not very clear to see except for the termites. I'm looking at the closed canopy and the open canopy there, and I think looking at the other colors at the bottom, they seem to be very similar. So when you get rid of the effect of the termites, I think that you might find that there's other interesting effects there that you haven't really discussed. And I think this is a problem because you've lumped everything under macrofauna. These animals, these are all different animals. I mean, your termites was just one species, so we can treat that as one species. You have other orders, and some of which comprise many species. But you've actually just lumped it all under macrofauna. So when you start looking at environmental responses and things like that, you're not necessarily getting the